meet or Zoom. Either way, if you have questions about like homework or about anything in the class. So give you a little bit background of myself. So I'm a chemist by training, and so in my lab we do a lot of organic synthesis. That's sort of our bread and butter, and but we also do like biochemical assays, cell assays, and also animal studies. So we sort of I always like to say we do like superficial biology, which help us to evaluate our compounds basically. Yeah, so the goal for us is basically develop chemical biology tools to help biologists understand their a system, or develop a drug, right, to treat certain diseases. So that's our research basically. So yeah, sorry, this is supposed to be updated, but anyway, I think you guys all have the schedule, right, from in Blackboard. Actually, I updated the one in Blackboard. And so for grading criteria, we're gonna have A, B, C, and F, right? 7 points based on attendance, 1 point for each lecture, um, and 28 points based on homework, 4 points for each homework, due 5 business days after the lecture, and 65 points based on the final exam. So if your final score is about 85, you got A. 70 to 84, you got B, and so on, right? Attendance is mandatory. I know like 2 students email me about they cannot make the the whole 2 hours of one lecture, which is okay, right? And for me, as long as you tell me, and if you make part of it and also you go back and watch the recording, that's okay. For me, I just want you guys to learn something, right, from the course. um, I can tell you actually, last year, for all the students, we only have one student got a B, and all the others got A. But that doesn't mean you don't need to work hard, right, to get an A. I'm just telling you, I'm giving you B is not my goal, right? I like everybody to get A, but I always like joke, I don't make more money for giving you a B. My goal is to get everybody understand chemical biology, and hopefully it will be useful for your future work. That's basically the goal for this course. And last year actually this course was very well received. I was very happy actually this course was voted by students as the best course in the CPSB program. And hopefully all you guys enjoyed this one for this year. Okay, yeah, I mean the final score may be curved based on students' performance, but I didn't need to do any curve. Only if nobody got A, I'll curve it. Otherwise, I think we don't really need to curve. Just to be more specific, I give a curve, the equation for the curve score. Because I got trouble before for students saying, oh, you are not like clear with the grading, so on and so forth. And I just want to cover all my bases here. Okay, so let's get started about chemical biology. So it's basically broadly defined as the application of chemistry method and techniques to the study of biology phenomena, right? So I think the key here is, if you look at this term, is biology, right? So it's not chemistry. Chemistry is basically the tool. Biology is your goal. So we try to seek new insights into biology by means of approach originating from enabling chemistry toolbox. You know, a lot of times I'll show you some examples, you know, why chemistry is very important in biology. And a lot of times you have to have chemistry to achieve your biological goals. I mean, the easiest example, the simplest example will be medicines, right? And a lot of the medications are small molecules, and they are, you know, from chemistry, right? From chemical synthesis. Without those, probably our lifespan would be way shorter than you know, what we have right now. So the you know, the objectives are, that's a typo, is not to for chemical biology, it's not to develop drugs, right? Although results obtained with chemical biology may well become useful in drug development. So you know, it's basically when we develop a drug, for example, right, for a certain biological target, if it, you know, if it works very well in both the cellular assay and animal studies, hey, we call that a drug, right? And we can further push this, you know, develop this. But if we cannot make it work super well in animals, we just call this a chemical probe, and you can use this to study your, you know, cell culture studies. But the important thing here is you want to make sure the probe or drug developed is specific, right? I think, okay, maybe let me step back a little bit. For chemical probes, you want this to be specific, because if you say, if you hit multiple targets, that will complicate your interpretation of your results, right? But as a drug, sometimes, you know, I would say it all depends, right? Certain drugs are, they call that multi-specific, right? Or so-called polypharmacology. It can hit multiple targets, and when you hit multiple targets, the outcome is better. The therapeutic outcome is better than just hitting one, you know, of those many targets. So I think the philosophy is a little bit different for drugs and chemical biology probes. Okay. Yeah, I I think I mentioned briefly in the Foundation A course, so I'll just reiterate a little bit in terms of applications of biology, chemical biology. You know, so there are many examples actually in the literature in terms of chemical biology, actually regulation of gene expression. Like hormone, they are small molecules, right? They can regulate gene expression. Actually, you know, that's a chemical biology process, right? So use a chemical to regulate the biology. And you can also use the you can also regulate the protein expression and functions. For example, you have a kinase inhibitor, you can inhibit the specific kinases. Like, you know, in bacteria, you can also use, you know, beta lactamase inhibitors to inhibit the beta lactamases in bacteria. And also you can use chemicals to regulate metabolic pathways and patterns, right? You can also use small molecules to visualize certain biomolecules in living cells. For example, if you have a, if you want to study calcium signaling, you can use a chemical, like small molecule based fluorescent probe called Fura2. You can basically treat your cells with this chemical probe, and then once you have calcium spikes, you can see fluorescence, right? Changes from the probe. So then you know, oh, I have calcium spikes. And you know, for the same reason, actually, you don't have to use small molecules. You can also use other engineered fluorescent proteins. You can also develop a calcium probe that way too. So I would say, you know, the chemical biology concept is very broad, right? So I think mostly we focus on small molecules, but you know, sometimes you can also use engineered protein to use that as a probe or sensor to evaluate the biological system. That's also broadly considered as chemical biology. I think this is a very loose term, right? We don't have to define exactly, what chemical biology is. Basically, the goal is to solve your biology problem, right? So, and also chemical biology can help you to identify binding partners of biomolecules and small molecule drugs. For example, like the drug example I just mentioned, some drugs have multiple targets, right? So you think, hey, for example, like the most simple example would be like kinase inhibitor, right? So in my lab, we work on like BTK, Bruton tyrosine kinase inhibitor. So that's a very important protein in the B cell signaling pathway. And there's a drug approved called ibrutinib, and everybody thinks hitting BTK is the target, right? Because they developed that drug for that target. Actually, I think that's true. But then at the same time, this drug can hit some other kinases too as off target, right? So how can you figure out these off target effects? Definitely, you can do a so-called kinase panel screen. You can send your compound out to a CRO company like Eurofin, right? So they specialize for this kind of kinase profiling. So basically what they do, they have these purified kinases, and then they have biochemical assay to see which your compound bind, right? Which kinase your compound can bind. So you just do that biochemically. On the other hand, with the new technology, with the development of mass spec, what you can do is you can use the so-called chemoproteomic strategy, right? So you can treat your cells with your drug, and if you can also incorporate a photoaffinity group, right, to the drug and shine light, and so the drug will cross-link to the protein binding partner, right, covalently, so stuck there. And then you can do pull down and run mass spec to see exactly which protein actually covalently bind to your drug molecule. Damien gonna cover extensively that chemoproteomic strategy in his lecture, and also we have Sanyun gonna talk about the proteomics in general, right? How it's gonna be useful for your research. And if you understand the off-target effects, then yes, then you will understand the potential toxicological effects, right? So you may, like, you develop a drug, you want this to be safe, and if you hit some important, say, housekeeping genes, and that could be bad, right? So you can basically identify liabilities early if you use this kind of profiling technique. So anyway, yeah, chemical biology is, I should say, it's gonna make your biology studies more convenient, and also you can reveal a lot of interesting phenomena compared with genetic strategies. So here's basically a brief summary of methods for studying protein functions. Definitely, I think everybody is very familiar with genetic knockout. You can do, especially nowadays, you can do this CRISPR-Cas knockout, right? Which becomes a kind of like a gold standard nowadays to develop mouse models. And you can also do this conditional knockouts. And also using RNA interference to knock out a gene, right? On the other hand, you can use chemical biology approach, basically use small molecules to turn off protein functions. So all these strategies, basically the gene knockout, conditional knockout, RNAi, basically they are pre-translational, right? Because that's basically you get rid of the protein before even they are formed, right? So you either get rid of them from the genome or get rid of them at mRNA level. But for chemical biology, that's post-translational modulation. Okay. So for genetic methods, right, versus chemical biology methods, I hope you guys have a good understanding of this slide, right? So because this is, I would say, important, because this is sometimes when you have certain, say, certain things you cannot achieve with genetic methods, then you should start thinking about chemical biology, right? So yeah, when I tell you guys important, that means it's important, right? That means probably it's gonna show up in your exam. Um, I just want to make sure you guys understand the most important thing from the lecture. And sometimes I give you some sort of more like introductory stuff. You can, listen to me, or you can sleep, either way, right? So that probably will not be in your exam, but probably going to help you to understand the topics better. I'll point that out. If something tell you, hey, this is not, you don't have to remember this. I'll let you know. Also, feel free to interrupt me if you have any questions. Okay. So for genetic methods, so what you do is you permanently shut off a gene, right? But that may cause some secondary adaptations in the mutant cells, because you get rid of certain gene, maybe other genes going to be upregulated to compensate the loss of that target gene, right? So leading to changes in expression levels of other genes potentially. And also viability could be an issue, because if you knock out certain essential genes, you may kill the cells, right? And also for genetic methods, it's mostly on off switches. You either have or you don't have, right? You could argue with RNAi, you can, try to dose change the dose of the siRNA and try to modulate the dose response a little bit, but it's not that easy to do. And also it's relatively slow, RNAi takes hours or days, right? And a lot of times we do at least 24 hours incubation, something like that. And also you manipulate gene or protein as a whole, right? Because you get rid of the whole gene, right? If you do knockout, you knock out the whole protein. You do RNAi, you degrade the whole mRNA, right? But in contrast, for chemical biology, right? So it's a short-term inhibition using a chemical only produces primary effects, right? So because the time course is so short, cells don't have time to adapt to the inhibition of that target. So this is good, right? So this is make your interpretation much easier because you're only looking at the primary effects. And also chemical inhibitors can achieve partial and continuous inhibition of your target protein, right? Because you can always titrate your drug, right? So if you know the IC50, you can, if you dose at the IC50, inhibit only 50% of the target, right? You can also play with the concentration to inhibit like 90% or 10%, right? So you can get a nice dose response effect. Or if you give a very high concentration and you can wash it away later. So if you do that, you can achieve a complete but transient inhibition even for target gene, for essential genes, right? So you can preserve viability that way. So that's a big advantage of these chemical probes. Yeah, so we say this continuous variability. So that's basically the dose response, right? By applying different amounts of inhibitor. And it's fast. You just throw them into cells, right? Most of small molecules get into cells pretty fast, within like 30 minutes. It can shut off the activity of target within seconds, right? A few minutes using small molecule as an inhibitor. So and another thing is, for example, you have a multi-domain protein. For example, you have a kinase, right? So you may have the kinase domain, say on the C terminal, some other, like domains on the N terminal. And sometimes these proteins have like scaffolding functions, right? So for example, kinase may act as a kinase, as an enzyme, but the protein itself can serve as a scaffold and bring together other proteins and form a complex, right? So these are two separate functions. If you do deletion, like genetic methods, you abolish both kinase function and the scaffolding function. Sometimes it may be good, right? But if you only want to understand the kinase function of that kinase in cells, if you delete the whole thing, it may not be good because you are confounding both the scaffolding function and the kinase function, right? But if you do this kinase inhibitor, you only inhibit the kinase domain and you do not affect this scaffolding function. So that's why we say for chemical probes, you can only perturb the targeted subdomain, right? If you have a multiple domain, like multiple enzymatic sort of domain, and you can develop different inhibitors for different domains that way too. Anyway, that's the advantage of chemical probes, right? But definitely there, all the methods or everything has its own pros and cons, right? So same thing, for everybody, right? You have your own strengths and weaknesses. So we have to be clear, right? What are the pros and cons? So for genetic methods, it's very specific, right? It's highly specific. It's well established, highly robust. You can essentially knock out any genes, right? Study all the proteins. It's very robust, right? And they can also routinely adapt in animal models. That's how we generate all these mouse models, right? Knockout mouse models. Baylor is very good at this, right? So we have a long history of generating mouse models. On the other hand, for chemical biology probes, it's very difficult to identify chemical biology probes that are specific for a target. That's why, I said, for small molecules tend to have potential off-target effects. Right? This is very important, right, to understand this, because if you use a non-specific probe, right? So although it can hit your target, it can hit some other genes too, and you see a phenotype, and you believe your probe is that protein is responsible for the effect, but maybe you're hitting other proteins which is responsible for your phenotype. So that just complicates your explanation. And the availability of chemical biology probes is very limited. It's case by case, because each one, we need to, like chemists need to spend a lot of time to optimize a chemical and get it specific, get it cell permeable, get it potent, eventually get a good pharmacokinetic property, and can be applied in animals. Right, so frankly, there's a lot of work to this chemical biology probes development. Yeah, that's why a lot of times people ask me, hey, can you develop a probe for this or inhibitor for this or for that? To us, any of those is a big commitment, because if you want to work on like a probe or inhibitor, at least a few years, right? So that's why for me, I always select the target very carefully. But for me, I'm always I like to collaborate with biologists. If you guys have a well-validated targets, right? Yeah, if you do all these knockout and knockdown studies and well-validated target and demonstrate that's important target, then definitely will be interested work on those. to develop small molecule probes. Like I said, chemical probes have limited bioavailability, so they need to penetrate different cell types from different organs, and also, certain small molecules are species specific, right? Yeah, the easiest example would be, for example, cereblon, right? Cereblon is a important E3 ligase, and thalidomide is actually is a drug, basically anti-cancer drug. Take advantage of E3 ligase activity, and the thalidomide can degrade a few important transcription factors in those multiple myeloma cells, right? Cancer cells, and to achieve toxicity. But interestingly, when you do the same experiment in mouse cells, you don't really see any effect. The reason for that is because in the mouse cereblon, compared with a human, there's one single amino acid mutation. And because of that variant, right, so you abolish the binding to the drug. That's why all these chemical probes potentially could be species specific. So you want to understand, right? So why or how well the compound work in different species. So it doesn't mean you have kinase inhibitor working in human cells, it's gonna work in mouse. So you have to be very careful about that. So to overcome the the bioavailability problem, or generality problem for small molecule chemical probes, like ATAG or DTAG systems are developed. So what are these, right? So there are some other tagging systems too. So what they do is here, actually, they try to use small molecule to do protein knockdown, right? So this is our so-called PROTAC systems, proteolysis targeting chimeras. We're gonna cover these in the protein-protein interaction lecture. I think I also mentioned a little bit in the Foundation A course, and I got like tons of questions after the lecture. Seems the students are very interested in these PROTACs. These are definitely very interesting chemical modalities. They are very new, right? And people are still exploring them, how to best use these interesting degraders, because before we mostly develop inhibitors, but for example, if the protein has scaffolding function, then the best way to address that is to degrade the whole protein, right, instead of inhibiting. And sometimes you get different phenotypes for protein degradation and protein inhibition. So then that'll be interesting, right? So one of the targets we work on, actually, they get, if you inhibit or if you degrade the protein, you get completely opposite phenotypes, right? So in that case, you want a degrader, right? So for the ATAG, DTAG system, we will introduce them more in the later lectures, but they sort of a more general way to achieve this protein knockdown using small molecules. Yeah, and then if you're more interested in this, you should read this review. Actually, it's very nice review by Kevin Shokat. It was published 2007, although it's a little bit early, but it's recognizing, exploring the difference between RNAi and small molecule inhibitors. And for DTAG, ATAG systems, you guys can read these papers. And also, this is from ATAG from C4 Therapeutics. It's a company. All right. Any questions so far before we jump in? No? Okay. Hopefully everyone's still there. Okay. Um, okay. So I think then we'll just move on. Protein labeling techniques, right? So this is actually very useful when you try to understand where the protein is, right? Your protein interest is in cells. So there are definitely a few ways of doing this, right? So you can use so-called conventional methods. You can use antibody-based immunohistochemical staining, like we call the IHC, right? Biologists are very familiar with this. You do that in fixed cells, and following the protocol, if you get a good antibody for IHC, you can stain your protein. But the thing is, you have to fix the cells, right? And another way to do this is you can fuse a GFP, right? Fuse a fluorescent protein or other fluorescent proteins to your protein interest. So you get a fusion. So now you can look at your protein interest in live cells. And you can, basically do a lot of interesting experiments. For example, if you add a stimuli to cells, and you can see how the protein distributes in cells, right? For example, if you give certain trigger, the protein may transport from the cytosol to the nucleus, right? Things like that, and you can quantify with this kind of fusion proteins. I think for most of the students in this class, at least based on my experience before, they are very familiar with these first two conventional methods. However, there are other methods, for protein labeling. I think is less familiar for the students. So one is so we call this the hybrid methods, right? So it's more like both I would say protein engineering and the chemical based methods, right? So it's a little bit complicated, but it has its own advantages. So one is based on the so-called single turnover enzyme based ligation. The other is using a peptide targeting sequence, and another way is to incorporate unnatural amino acids, right? So this is important slide. I hope you guys at least know the general category of these labeling methods. So what is the single turnover strategy, right? So Halotag technology developed by Promega. This is the one actually we really like. We use it a lot in the lab, and we basically. So what is this strategy? Protein tag, right? Like a Halotag is a modified haloalkane dehalogenase, right? Haloalkane dehalogenase designed to covalently bind to synthetic ligand. So what does it mean, right? It's not easy to read here. Basically, what it does is, this protein, basically, the job of this protein is to remove chlorine from a molecule, right? So this is actually a bacteria protein, right? So they stay in like very dirty environment. Sometimes they don't like to have this chlorinated compound in their system, so they want to remove this chlorine, right? And basically make a Cl minus, like chloride, and then they can hydrolyze this. Basically, this become a OH. So they replace the Cl with the OH, right? And then so they remove chlorine from the molecule. That's what the enzyme does. Basically, what Promega did was they developed a mutant, right? So basically, this reaction is a two-step reaction. So there's an aspartic residue inside the cavity. That's the active site, right? So they use that this carboxylic acid group to attack this chlorine and kick it out, then they need to hydrolyze it, right, and release this molecule. But they stop the reaction. They introduce a mutation, basically only allow the attack of this chlorine, but doesn't allow the enzyme to hydrolyze it, right? Essentially, if you give this chlorinated compound, this compound gonna stuck on this enzyme, right? So we call this single turnover enzyme. Um, so what's the advantage here, right? So basically, if you can imagine if you put a dye on this chlorinated compound, right? So once this compound interact with this single turnover enzyme, this enzyme going to be labeled by this compound permanently, right, covalently, and that's it, right? So then um, um, this protein become fluorescent because you have fluorescent dye on here. And this process is actually very fast. That's why we really like this system, like the Halotech system, because the reaction is extremely fast. You generally only need a few minutes to achieve this labeling. And also the structure of this tag is very simple. As a chemist, we love simple structures because we can easily, play with the dye structure, right? So, and then you may ask, hey, why you really wanna adapt this system since this is so complicated, right? So if you think about this, if you want to label protein interest, you can just fuse the GFP or RFP to your protein interest. Then you don't have to do any of the labeling. You can visualize them in cells, right? That's completely true. The downside there is, say, you label your protein with GFP, but then next time you want to label, maybe say you use a dye, also use a small molecule dye, also using the GFP channel. You want to do the experiment simultaneously with your protein tracking. Then your GFP fusion protein will not work because both the small molecule dye and the protein gonna show up in the same channel, right? So you're gonna get a whole blob right there. Then what you need to do is you need to make a new construct, right? So then you can put an RFP or any fluorescent protein there as long as it's not green. Yes, you can do that, but definitely, you need to make a new construct. You need to do cloning or outsource this cloning, so on and so forth, right? So it takes some time to do that. But on the other hand, if you use this Halotech strategy, what you can do is you can just make one construct, right? And you can just make this halo tag protein fuse with your protein interest, and then express this, right? And you can either transiently or stably express this protein in cells. Then you can, just depending on what kind of dye you feed to cells, and your protein interest is gonna be labeled with the different color of dye, right? So just a matter of put a different color of dyes in cells, right? So this just makes things very convenient, right? Especially when you want to switch colors. Another thing is these small molecule dyes generally have way better quantum yields and photostability than fluorescent proteins. And that will allow you to do long-term tracking, right? And because of less photo bleach. And also it can provide certain wavelengths which may not be available using fluorescent proteins, right? So there are definitely many advantages of this. And also you can do the so-called pulse chase experiments, right? For example, you want to measure the protein half-life of your protein interest. And then what you can do is you can express this Halotech fusion protein in cells. Then you add this small molecule probe to cells. You wash it away, right? So basically, all your proteins gonna be labeled Halotech protein, right? Protein interest is gonna be labeled with the dye, but then you don't have free dye anymore in the cell culture. Then you just wait, right, and do imaging and look for the degradation or disappearance of the fluorescent signal, right? So because once protein get degraded, and the dye gonna become small molecule and it's gonna diffuse, gonna come out of cells too. So you start to lose fluorescent signal. So that's a way actually to track the protein half-life. So we call that pulse chase experiment. On the other hand, if you think about GFP, that'll be hard to do, right? Because it's continuously expressed unless you inhibit the protein synthesis, but then that will cause some other issues too, right? So it's not as ideal as this pulse chase halo-tag experiment, right? Definitely you can measure protein lifetime with other strategies. For example, mass spec strategy, you can pulse chase with other like heavy isotope labeled amino acids, and then do experiments like that. But that'll be a different story, right? So I'm just talking about one protein interest and doing it conveniently using a microscope. Okay, definitely there are other competing strategies for this single turnover enzyme. There are other strategies called the snap clip strategy developed by Kai Johnson and Kai Johnson group. I mean, frankly, we seldom use this because number one, if you look at this, you have to incorporate this structure to the fluorescent dye, and the synthesis is more challenging here, and also actually because they use this kind of nucleotide structures too water soluble, so the permeability of this conjugate is generally not very good. And also the labeling kinetics is slow, right? So my advice is if you're gonna only use one this single turnover enzyme, go with halo-tag, right? And if you want to do two, then probably have to pick a halo and snap, basically something like that. But I think as long as you guys know this is out there, that's good enough. But halo is the preferred method. Okay, but then what's the disadvantage of this halo-tag and GFP, right? For example, like GFP and halo-tags, they are all big, right? So they are big proteins, and a lot of times you have to assume with that big protein you don't disrupt the biological functions. Some, I would say most of the time it's true, but sometimes you have to be very careful, right? Especially when you have protein-protein interactions. You have such big GFP there that could be troublesome. So that's why in the field, in the protein labeling field, people are always thinking about how can we minimize the tag, the size of the tag, right? So to address this problem, actually, Roger Chan, one of the key players in the GFP field, right, who won Nobel Prize, he actually came up with so-called FLASH or REASH system. This is a sequence of this tetracysteine, right? So you have cysteine, two random amino acids, and cysteine, right? So you have a tetracysteine sequence, only 6 amino acids. You can encode that to either C terminal, N terminal of the protein, and or even insert within the protein, right? So then this tetracysteine sequence can be specifically recognized by membrane permeable fluorescent derivative with 2 arginine substituents, like for example shown here. That's called a REASH. So the good thing is, this molecule is non-fluorescent, right? So when you have this as a small molecule, so when it binds to this tetracysteine moiety, it will become fluorescent. So that's cool, right? So because this is the turn on probe, for example, like the halo-tag strategy, if you just use a conventional halo-tag substrate, if you didn't wash the probe cleanly, and you may have free probe in cells, right? But that doesn't reflect your protein location. That will, mislead your result. But definitely there are so-called turn-on Halotech substrate out there, more like in the developmental stage. I don't know whether they are available through Promega, but they are available based on literature. But here, this is a turn-on probe, right? So even you don't do cell washing, these compounds are non-fluorescent. Once they bind to your target, they become fluorescent. So that's a nice design. But to tell you the truth, this system, although it's was developed say early 2000, it's not widely used, mostly because this arsenic compound actually has toxicity, and the labeling is not very specific, basically. Although they think this tetracysteine moiety is the only moiety the molecule binds, but actually this compound can bind non-specifically to other like cysteine residues in cells. So that's why this is more like, I think, let you guys know this thing is out there, but this is not something I would say commonly used in research. Okay, then another interesting development is from Alice Ting's group. Alice was Roger Chen's postdoc, and she's also one of the major players in the protein labeling field. What she did was actually pretty smart. She's trying to take advantage of these ligases, right? So they call this biotin lipoic acid ligases. So what they do, what she did was basically you just need a short peptide sequence as an acceptor sequence. You can encode this very short peptide, like about 10 amino acids into the protein, and then these ligases, right, engineered biotin or lipoic acid ligases, they can attach a bioorthogonal chemical groups to the protein. Then the protein can be labeled with different dyes, right? So it's a two-step labeling. So for example, in this biotin ligase case, basically the job for the biotin ligase is to attach, this biotin residue, or say this small molecule, right, to the protein, right, through forming an amide bond, right, with the acid here. But if you replace the biotin with this ketone, right? If you look at here, what happens is basically this is you replace the NH with carbon, right, with CH2. If you just look at them, they look very similar, right? And that's why if you sort of trick cells with this ketone, and this biotin ligase can still recognize this ketone, although the affinity may be lower, but it still can accept this substrate. And then here's the thing, right? So if you have say this is the extracellular, right? So you express a membrane protein with this tag, acceptor tag, and then with biotin ligase with this ketone 1, and you can attach this ketone to this acceptor peptide, right? Then because ketone is a reactive moiety, and we can do a lot of chemistry on ketone, but we cannot do chemistry, or say very difficult to do chemistry on this so-called urea molecule, right? So with ketone, and then what you can do is you can take this so-called hydrazine, and this one can form a very specific covalent bond with ketone, right? You form this kind of structure, right? So you can imagine similar to the Halotype example. So this is more like the chlorinated part, right? And this is the dye. And then you can basically attach the dye to this ketone, right? And introduce the fluorescent molecule to the protein. So yeah, this is basically how you can do this in a two-step manner. Yeah, so this is pretty I would say pretty interesting strategy. And Alice team has done a lot of work in this area. Actually, you can see she started like 15 years ago. And then there are multiple iterations of this, not just for biotin. And later she started work on lipoic acids, try to basically the same thing, right? Lipoic acid structures like this. Sorry, it's just like from here. Maybe let me label. Yeah, lipoic acid is just like this part with OH here, right? And but for this lipoic acid ligase, basically what it does is just the same thing, right? Form a covalent bond, amide bond between the lipoic acid and the protein, right? So you attach this lipoic acid to the protein. And then what she did was basically similar strategy. Instead of having lipoic acid here, you can put a azide, this N3 group called azide, right? And then you use this azide group. So basically this as a substrate, right? And then you can basically incorporate this azide to your protein specifically, right? With this lipoic acid acceptor sequence. And then what you can do is you can do this so-called click chemistry, which we'll briefly talk about in the next hour. So basically, if you have this kind of alkyne, right, the triple bond, and it can specifically react with this azide, right? So this then you can essentially label this. You can put this a dye in the red part or biotin for pull down or anything, right? So you can think about. So you can basically you just introduce a specific handle to your protein interest. Then you can modify with this click chemistry, and you can modify with anything you want, right? So that's a pretty cool strategy. Right, similar concept to the biotin ligase. The difference here is for I think for the biotin ligase is now cell permeable. The ketone itself is now cell permeable, but this one can be used for intracellular target. Yeah, that's why. Why I cannot proceed slides? Okay. Yes, that was the lipoic acid, right, labeling. There recently Ting's group developed some other strategies called proximity labeling. The idea here is it's more like wanna study protein-protein interactions, right? So when you have very weak protein complexes, especially very transient protein complexes, you want to see which protein actually interact with your protein interest. If you have weak interaction, it's difficult to do pull down because you need enough affinity, right? For example, like nanomole, micromole, at least affinity to your target protein. But if your binding is very transient, very fast, it's difficult to catch those. So what Alice Ting did here is basically she used a for example this you know, the peroxidase, right? And they call this apex. Um, so basically, the idea here is you can fuse this protein, right, to your protein interest, like they show here, right? And then what they do is this enzyme, this mutant enzyme, whatever, you know, this enzyme can basically catalyze the reaction, basically, you know, convert this phenol into a radical, you know, like here. So this enzyme can convert this non-reactive substrate into a reactive one. And this, because this is a radical, so it has very high reactivity, essentially just going after whatever protein next to it, right? Will just react to it. So if you're close by, this radical gonna react with the close by protein that way. Then if you say you have protein very far away, but during the you know, diffusion process of this radical, it's going to be quenched by solvents, right? And so you just lose the activity of this radical. And you know, you can also introduce a biotin to this phenols. We call that biotin phenol. So basically, all the proteins in vicinity going to be labeled by this biotin phenol radical, right? Then you can do structure evident beads pull down. Then you can send this to mass spec to identify what kind of protein you have in the vicinity of your target protein. So this is called proximity labeling. They have many iterations now. I think this is a very hot area, basically to study protein-protein interactions. So I hope, you know, if you guys interested in this, you should check out Alice Ting's paper to understand the protein-protein interactions. And also, we can introduce so-called unnatural amino acids into a protein specifically. This was pioneered by Peter Schultz at Scripps. You know, this is basically they take advantage the codon not used by protein translation. So then they develop a orthogonal tRNA. You know, basically to introduce unnatural amino acids to those codons, and then you can specifically introduce a natural amino acid specifically in the protein, right? So this is, I think, this technique is very specialized. There are many orthogonal tRNAs being developed. You know, Dr. Wenxue Liu at Texas A&M was a postdoc in Schultz group. He is the local, I would say, local expert for this type of natural amino acid protein labeling. Definitely, I think other groups are doing that too. At Baylor, mostly apply this technique for you know, like protein expression, protein labeling, or even in structural biology studies, right? But anyway, I think for you guys, I think as long as you know, there's a strategy called incorporation of natural amino acids, and to label protein specifically, that's good enough, right? So in the future, if you really want to apply this, you need to talk to some of the experts, not me, right? So we don't really do this. All right, I think that's good for now. Maybe we can, I guess everybody probably tired. We can take a like 5-minute break, and we can come back and finish the second part of the lecture, which should be short. Let's come back like 12:05. Okay. Okay. Oh, there's a chat. Okay. What is it? Oh, I guess okay for taking a break. Okay. Oh, right. Let's finish this. I guess everyone is hungry. I don't know why they chose this 11 to 1 p.m., but hopefully you are still alive. Let's do this quickly. Share screen. All right, I think we are good, right? Can see my screen. Okay, great. Kim is recording this. Okay, so next one we are gonna talk about briefly about this bioorthogonal reaction. Okay, yeah, I'm listing a lot of different reactions here, but don't be scared. I don't expect you to know all these. I think for you guys, the most important thing is, you got some basic ideas. I'll let you know what you need to know. For biomolecules, they have like different functional groups, right? For example, you may have cysteine, right, in a biomolecule, and then you can label label that cysteine with a chemical, right? And that's also one strategy people use in drug development. They develop this so-called irreversible covalent inhibitor or reversible covalent inhibitor, right? And there are other chemistry developed to target a specific lysine. I think that those are interesting things to explore. But in terms of bioorthogonal, basically what they want to do is they don't want this chemical react with cysteine or lysine or this commonly observed, commonly appeared in protein molecules, right? Common functional groups in protein molecules. What they want to have is basically, a group which generally does not exist in biomolecules, right? And then you can have a reaction specifically to that group. The easiest example, like in Alice Ting's study, right? So you can introduce an azide N3 group to a biomolecule, either through the acceptor tag or through the natural amino acid incorporation, right? And you can introduce the azide to that biomolecule of interest. Then you can run the so-called click chemistry. You form a reaction between this azide and alkyne group, right? Then you can label this with any dyes or biotin for pull down, things like that, right? So there are different types of bioorthogonal reactions. We don't have to go through all these. The most important one is we call it 3+2, which is the click chemistry, right? So we use this very widely, like the azide alkyne chemistry. Yeah. I guess we can just skip this. This is too much for you guys. This is also too much. But what we can focus here, we can just focus on the first line here, right? So I think that slides I put in here is mostly for your information, right? In the future, say you want to use this bioorthogonal chemistry, but then you say somehow you cannot use azide alkyne pair, or say that's already been used in your system, you want to have two kinds of bioorthogonal chemistry, then you can come back and check this table, right? See what others available here. But I mean, for our purpose, right? For our course purpose, I think as long as you guys know this N3 azide can form a specific reaction with this triple bond, right? Alkyne bond. That's good enough for me. Okay. So I'll tell you like two different versions of this so-called azide alkyne click chemistry. So one is the traditional one called copper catalyzed click chemistry, right? So we call this. And one thing you guys want to remember, this is a copper 1. It's not a copper 2. So what is copper 1, copper 2, right? So copper 2 is the common ion we see for copper, right? So if you see the nice blue when you have copper sulfate, that's actually copper 2, right? But that's not the catalyst for this azide alkyne reaction. The catalyst is actually a copper 1. So you have to reduce copper 2 into a copper 1, which is colorless, right? So sometimes, and also the copper 1 sometimes has a relatively poor solubility. So you can see some precipitate when you reduce the copper 2. That's very normal when you run this type of reaction. So don't be surprised, I have precipitate, the probably my reaction is bad. No, that's a good sign. You have reduced the copper 2. So you have that, then that copper 1 can catalyze this azide alkyne reaction, right? And you can specifically label protein this way. But definitely there are caveats for this, as you can think about, right? So copper is not very so-called biocompatible, right? So it can cause cellular toxicity and cannot be applied in live cells. So this reaction is very commonly used. Let me change my cursor. This reaction is very commonly used in like proteomics, because you basically you kill the cells, right? You get lysate and you try to do pull down studies. You don't really care, right? And so this copper catalyzed click chemistry is commonly used. But on the other hand, you cannot do this in live cells. You're gonna kill cells. What you can do with live cells, you use this so-called strain promoted azide alkyne cycloaddition reaction, right? Or SPAC. So what does it mean here, right? So if you guys still remember the sophomore organic chemistry, what's the bond angle between this and this, the bond angle of this alkyne? Anybody knows?
Is it 180 degrees? Like it's just flat?
Yes, exactly, right? Yeah. So the bond angle here is 180 degrees. Okay, so then if you form a ring here, what happened is, can you achieve 180 degrees if you have an 8-member ring?
No.
Yeah, I would say it's gonna be very hard, right? So it's sort of you have to bend the molecule somehow. If you have a super large ring, then that's probably it's okay. But with this a small ring, actually, it's very hard. So that's why you bend this triple bond. You put a lot of strain, right? It's not in a comfortable position for this alkyne. So that's why this alkyne becomes very reactive towards azide, right? So that's why they call it strain promoted azide alkyne cycloaddition reaction. Yeah. Then you may ask, hey, why not just do 7-member ring or 6-member ring? It's even more reactive. To tell you the truth, those compounds are not stable. You can actually make those as a so-called reactive intermediate, but it cannot stay in a while. Basically, you cannot just use it conveniently, right? So you could generate that as a reactive intermediate, which can disappear within less than a second or something like that. So that's why they just settle with this 8-member ring. And you actually, because this was developed by Carolyn Bertozzi, now at Stanford, when she discovered this was when she was at UC Berkeley. She made her name in this basically copper-free click chemistry. And so they basically you can apply these to cells, and you can, you know, if you can specifically incorporate the azide into cells, and then you can label the protein with this kind of string promoted alkyne. And a lot of this reagent is actually commercially available, right? So it's just no metal catalyst. But the caveat here is you want to pay attention to is some of this alkyne, right? This 8-membrane alkyne can react to thiols because they're just so reactive, and you know, sulfur can react with this alkyne. That's actually a potential, I would say, a downside of this, right? It's in terms of specificity, it's not as good as this copper catalyzed reaction, but you know, definitely copper catalyzed cannot be applied in live cells. So you just, you know, my point is all technologies have its own pros and cons. You wanna be aware, right? So what potential complications you're gonna have. For all these, we don't have to know, like I said, I mean, you guys don't have to know, as long as, you know, they stay in your PowerPoint, you can always go back and check them. Yeah, again, these are also some of the interesting so-called Diels-Alder reaction, and they have interesting applications for certain you know, area, but again, you know, you don't have to know this. Okay, so how can we apply these bioorthogonal reactions, right? So this is just a I would say simple example, right? So what you can do is, for example, if you have a cell, so you have many proteins in there, right? So what you can do is you develop a say irreversible covalent inhibitor, right? So maybe let me just draw on here, make sure you know, right? So you have a molecule, right? And the molecule has an alkyne on it, but then the molecule has a reactive group that can specifically react to your target protein, right? So that's called the activity-based probe. So that'll be good, right? So for example, like for protein tyrosine kinase, BTK, and you can develop an irreversible inhibitor, which can react you know, irreversibly with BTK. Then because you have a alkyne to your on the small molecule, so you essentially you label BTK specifically with this alkyne, right? And at the same time, you know, if this molecule has off-target effect, maybe, you know, it may react with other proteins irreversibly. You know, those proteins gonna be labeled by the alkyne also, right? So that's good. That's how you can profile all the off-target effects. For example, if you don't have this so-called activity-based probe, right? So you cannot form a direct, say, covalent bond with your target protein. What you can do is on the small molecule, you can introduce a so-called photoaffinity, PAL, right? So photoaffinity labeling group, photoaffinity labeling group. That's the I think that's the key in Damien's lecture, right? So for the chemoproteomics. Then what you can do is say, you know, now we don't have this anymore, right? So you have this photoaffinity labeling group. It can be a azide, actually it can be diazerine, you know, can be any of these photo activatable groups, and also has a high reactivity to label protein residues. So basically this part is a small molecule which can bind to protein, right? And then once you get the small molecule close to the protein, then you shine light, this photoactivatable group gonna cross-link to the protein, right? And so then once you have that, you definitely have a lot of proteins which are not labeled. And so what you can do is now you can run click chemistry. You can have a, this is biotin, right? So that looks like a biotin with 2 5-member ring fused together. So you can have a biotin-related azide. You can run a copper catalyzed reaction to label that protein. Okay, so then you can do strap evident pull down. You pull down this whole protein. And then you can do this so-called on-bees trypsin digestion. And you basically, you release, you basically get the, this protein chopped, right? And then you can release this labeled peptide, and then you can run LCMS, MS, right, to get the, and then you do informatics to figure out what protein is. So one thing is I wanna point out is, when you have this pull down, right, and you need to wash this multiple times to remove non-specific binding, right? So then when you do on-bead trypsin digestion, you actually get both. So basically, you chop this protein off, right? Because you digest it. So you will not only get the peptide connected to this azide part, you are also gonna get peptide from other parts of the protein, right? Through this digestion process. So this cartoon actually is not very good. You should have other red peptides in here too, right? So not only just release this peptide, you're gonna have other peptides. Then you run mass spec. You can identify not only the azide containing peptide, you can also identify other peptides from that protein. That will just enhance your confidence to identify that protein, right? Because you can match this part to that protein, also match that part, right? If you match multiple parts of that protein, hey, that's something I have. So that's one way of doing this. Alternatively, you can also, once you label the protein with alkyne, you can also do the digestion directly. And so then now you have all these peptides, right, in the system. Then you can do biotin, this biotin-related azide click chemistry, right, to label that peptide. Then you do pull down with the peptide. So here you want to pay attention, right? So when you do this strategy, you lose all these red peptides, right? Unlike here, you can have identification of this red peptide, which does not have azide. But in this strategy, you lose those. So you only rely on this labeled peptide, for identification. So each method has its own pros and cons, right? Because here, when you have these, protein pull down, like pull down the whole proteins are, more sticky than peptides, right? So you may have non-specific interaction here, a more non-specific interaction here, right? And but with peptides, you're gonna see less. But at the same time, you lose other non-modified peptide information from this strategy. So it depends, right? So your goal, right? Essentially, how you want to design the experiment. Sometimes, for this strategy, if you really want to understand the binding site of your molecule, this is a good way to do it, because you don't have other complications or other peptides, right? So you just directly look at that binding site of the peptide. But you want to just identify the protein, the identity of the protein, not just a binding site. This will be a better way to do it. Yeah, is that okay? Okay, clear all drawings. Okay, so that's you know, in here we basically use a small molecule basically bind to the protein interest and introduce you know, alkyne group or handle group to the protein interest. That's the one way of doing that. Another way of doing this is basically we call this metabolic labeling. Metabolic labeling is basically you take a, for example, like here is a sugar, right? So it's a glucosamine. It can be used as a surrogate of N-acetylglucosamine, right? So basically, for glucosamine, you just have NH2 here, but you know you put this little azide group on here. So because this group is small enough, you know, enzymes in cells still can tolerate this, can incorporate this N-acetyl acetylglucosamine just as you know, N-acetylglucosamine. So essentially, you know, you incorporate this azide labeled sugar into the sugar polymer on cells, right, in cells. So in this case, this glucosamine is displayed on the cell surface, and then you can basically with this metabolic labeling, you can display this azide on the cell surface, and then you can do click chemistry and label the cell surface with the fluorophore. So the concept here is very similar to you know, heavy isotope metabolic labeling, right? So you can throw in heavy amino acids into cells, and cells get labeled by these heavy amino acids. Those heavy isotopes are just a better mimic than these chemicals, because you only you know, you only add a neutron basically, right, to those elements. But here you have to introduce some sort of groups, right? You change the molecular property a little bit, but you know, the changes is small enough to be tolerated basically. So that's so-called metabolic labeling. You can do the same thing with proteins, right? So you can basically use a azeto aniline basically to you know, throw that in cells, and a lot of the methionine position is going to be labeled by those aniline. So you know, it's a similar stuff, right? So here just to show you a sugar example, but you can do the same thing with proteins. Okay, yeah, for lipid labeling, this is also a very hot area, I think, in the biology field. You know, trying to see how lipid composition, right? So for lipidomics, you want to figure out the lipid composition, and once you know that, you want to know, hey, you have, say, lipid A is you know, like upregulated, right? And you associate that with phenotype, but then you want to know the biological target of your lipid, right? So it has to work on certain proteins or biomolecules to exert its effect. So the way to figure out the lipid binding partner, you can apply a similar strategy, like I mentioned, this photoaffinity labeling approach, right? So this group, this triangle, right? So with two nitrogen called a diazerine, right? DIAZRI, diazerine, okay. Anyway, you can read this paper to get a little bit insight about this. So you can look at this, right? So they developed this probe, they incorporate this diazerine group as a photoaffinity group in there. Also, they have a alkyne handle on here. So yeah, this is they also try to position this photoaffinity group at different positions, like here, right, and keep the handle here. So yeah, there's a lot of chemistry you can play with. Yeah, I mean, to tell you the truth, actually, we made some of these probes following Ben Kravat's paper. These are very difficult to make. It took, I think, a postdoc like 3 months straight. There are like 20, 30 steps to make these, although they look pretty simple, but they're very, very hard to make. Yeah, that's why, you know, a lot of times this chemical biology type of thing is not widely used in among biologists because the limited availability of these probes. Um, yeah, so, you know, as a chemist, I'll be happy to help you guys if you have any help, you know, if you need any help. Um, and yeah, I think Damien also gonna talk about this example in his lecture. Another interesting thing, this is something I added like relatively new because there are a lot of interest nowadays to study RNAs. And you know, in biochemistry department, in our CPSB program, we have two new recruits, assistant professors studying RNAs, right, and the function and binding of RNA. So I just sort of throw this out there. Hopefully it will be useful for you guys. So for RNA labeling, you can do the so-called fluorescent in situ hybridization, the FISH method, and you can also use this so-called molecular beacon to do this turn on FISH. These are basically well-established methods, right? I don't want to go into details. But on the other hand, there are other ways, like relatively new ways to study RNA. They call this light up aptamers. So what you can do is this aptamer is basically a sequence of RNA. They call that aptamer. And this aptamer can specifically bind to this small molecule, right? So they can evolve this aptamer against this small molecule and make it specific, sort of like you raise antibody, right? So you can inject antigen to animal and they will produce antibody, or you can do this sort of multiple rounds of selection, like phage display sort of strategy, right? Try to get a high affinity antibody. This is the same thing. Aptamer, just antibody based on RNA, basically, right? And that's called aptamer. And the idea here is kind of like a GFP, right? So you can fuse this part of aptamer to any RNA of interest. So you get a fusion, and you can label that RNA, specific RNA, with this aptamer. And so then how does it work, right? So then you have this small molecule. Basically, generally, you know, this small molecule have two rings, and because of the free rotation, right? So you don't have fluorescence. They are non-fluorescent. Okay, so basically, if you apply this small molecule in cells, you don't see any fluorescence until this small molecule bind to the aptamer, and you lock these two rings in position, right? So they cannot do the free rotation anymore, and you lock them, and you start to see fluorescence. So that's a turn on process, right? So student may ask, hey, why, you know, the rotation gonna make fluorescent, I mean non-fluorescent? So the thing is, when you put energy into a molecule, right? So you make this molecule into the so-called excited state. Once the molecule is the excited state, it can have multiple choices to lose that energy. It can basically emit a photon, which is fluorescence, right? It can relax back to the so-called ground state, go back home, right, that way. Or it can, you know, do some rotations, whatever, waste the energy. Instead of emitting a photon, it can just do rotation, right, and sort of waste energy that way and relax back to home, right, to the ground state. For that process, you know, if you do the free rotation, apparently you don't emit a photon, so you don't have any fluorescence, right? So when you lock this molecule in place, it cannot, you eliminate a deactivation pathway, which is the free rotation, right? So if there are only two pathways to deactivate, then it has to emit a photon. So that's basically how this, I would say, how this thing works. Yep, that's all I have for today. Do you guys have any questions? Okay, if no more questions, I don't know why in this slide I don't have the homework, but let me see whether this. Uh, yes, here's the homework. Let me show you the homework. Yeah, can you guys see this? I think this is in the in Blackboard. If it's not, I can always send this to everyone. Basically, it's just try to apply one of the chemical biology techniques you learned today to answer a biology question in your current lab, right? It can be your current rotation lab or lab for your PhD. Or it can be previous rotation lab. It doesn't matter if it's too early for you to understand the projects in your current rotation lab. And this is totally open-ended. I wanna see how you guys understand what you learned today. For me, I just want to make this useful for you guys. And the goal is to help your research, right? Actually, last year I got very good experimental designs. Actually, they ask like certain interesting questions. They sort of design experiments, try to answer those questions. Yeah. Okay. If no more questions, yeah, I'll post this in chat. But if no more questions, see you guys next time. Bye.
I had a quick question.
Oh,
you want
sure.
the homework emailed to you, or is there a place on Blackboard we can submit it?
Yeah, that's a good question. Hey, Kim, what we did last time, I think you emailed to Kim, right? Or you can upload to Blackboard. Either way, I think.
I'm sorry, I didn't. I didn't.
Oh,
question.
hey, Kim. Lauren is asking like, how do we deal with the homework? Do they email to you?
Yes, the homeworks are submitted to me, and so I'll just keep track of, like who's turning in, and then after I receive everyone's homework, I'll submit to the instructor of the lecture.
Yeah, that's the way it works. Yeah.
Thank you.
Yeah, no problem. Okay, see you guys.